Okay, that was you. Okay, well, welcome on this nice Monday morning in rainy Honolulu. I'm very happy to announce one of my long-term friends and colleagues, who is Dr. Dan Chertow, who is a tenure track investigator at the critical care medicine department at NIH in Bethesda, and is also affiliated with the Laboratory of Virology at Rocky Mountain Labs in Montana. Dr. Chertow received his medical degree from Northwestern University Feinberg School of Medicine and a master's degree in public health from University of California at Berkeley. He's actually a public health service officer as well, and completed medical internship and residency at UC San Diego, and then a fellowship in critical care medicine at the NIH and infectious diseases at Johns Hopkins. Dr. Chertow attends in a multidisciplinary intensive care unit at the NIH Clinical Center and does translational research on the pathogenesis and treatment of severe emerging viral infections with the colleagues at Laboratory of Virology. Of note is also that Dr. Chertow was participating in the Ebola outbreak response during the West African outbreak. And so we also know, of course, of his work in the field of filovirology, but today he's going to be talking a little bit about COVID. And without further ado, please.
Right. Okay. So thank you so much for the kind introduction, Axel and Vivek. I appreciate you having me here, and I'm excited to talk to you all and to share some of our work around COVID that we've been focused on for the last few years with a large collaborative group. So the title of my talk today is A Paradigm Shift, Widespread SARS-CoV-2 Infection and Persistence in Humans. And the shift really is this concept of thinking about CoV-2 now, not just as a virus that infects the lungs, but in fact is a virus that can disseminate throughout the body and infect cells and tissues throughout the body, and perhaps even persist in those cells and tissues for the long term. So that's sort of the bottom line up front, and I hope to convince you of that as I go through my talk. Okay. I have no disclosures. When we started this project, we really had 4 objectives. The first was to describe the burden of SARS-CoV-2 infection, both within, but importantly also outside of the respiratory tract. And the second was to characterize the cellular tropism of the virus, both in the body and including brain, to understand, at the cellular level, which cell types were actually infected. And then the third was to determine if either live virus or overall components actually persist in tissues, and if so, over what time interval. And then our last objective was to assess whether CoV-2 replicates in those different anatomic compartments, and if it is replicating, is it actually evolving within the host over time. So with that in mind, when this started, we collaborated with 7 hospitals in different regions across Maryland. A number of these hospitals were affiliated with the University of Maryland, and then also Tidal Health. Among those hospitals, we had contacts. I think Axel mentioned I'm an ICU doctor. So we worked with colleagues and collaborators that worked in the intensive care units of these hospitals at the beginning of the pandemic, such that if they had individuals who died, they would ask the next of kin if they might be interested in participating in our study. And if they were, I would be in touch with them, and often in the middle of the night, we would consent them for study participation. And then we would work through the admissions department at the NIH Clinical Center to facilitate transfer of the body or the remains from the hospital of interest to the NIH Clinical Center. And this would happen sometimes in the middle of the night, so that the next morning, my team, my laboratory team, in conjunction with the really remarkable pathologists within the National Cancer Institute, we would be staged and ready to do these autopsies and the tissue procurement. And our goal was to do all of this within a 24-hour interval of death, to really try to reduce that postmortem interval to optimize the amount of information that we can get from these individuals. And so we ultimately performed 44 autopsies. These took place during as I mentioned, the first year of the pandemic, from March of 2020 to March of 2021. And this is the epidemiology curve of cases in Maryland during that time frame. That's the blue line, and the overlaid red arrows are the autopsies that we performed. And so you can see that we captured deaths across the multiple waves and pretty well distributed throughout that first year of the pandemic. During these autopsies and tissue procurement, we tried to optimize sample procurement so that they were amenable for the different downstream analyses that we were planning on. And they really sort of, the tissue procurement sort of fell into three categories. So one, from any given tissue, we would collect a sample and preserve it in RNA later. So that was amenable for downstream molecular analyses, whether that be PCR or sequencing or otherwise. From an adjacent piece of tissue, we would collect a little piece and flash freeze it, so that subsequently, if we wanted to go back and look for viable or live virus, we would have that available to us. And then the third would be to take a piece and then to fix that piece. And, you know, working with our pathology colleagues, the fixation approach that we took was designed to be amenable for detecting nucleic acids and proteins within tissues. And it's another way of saying that we intentionally did not overfix these tissues with formalin that would reduce the likelihood of picking up RNA or protein. They were in formalin for 24 hours and then moved into 70% ethanol for the purpose of trying to preserve nucleic acids. So if a signal was there, we would pick it up. And then I'm just going to go in and describe our cohort. So we had a pretty diverse cohort. You know, the average age was about 60 years old. But if you look at the range there, we had folks that were as young as 6 years old and as old as 91 years old. Had a pretty good ratio, about 70% male, but 30% female. And then a reasonable diversity across race and ethnicity, with about 41% individuals who identified as Black or African American, 41 as white, and then another 16% that identified as Hispanic or Latino. It's important to note that among our cohort, as I mentioned, this was done during the first year of the pandemic. And I'll just highlight now that at that time, vaccines were not available. So nobody in our cohort was vaccinated. And mostly, you know, as pointed out, you know, these were older individuals and individuals with multiple comorbidities. They were hospitalized often from or with COVID, you know, severe COVID. And so there was a high level of comorbidities, and about 77% of our cohort had 2 or more comorbidities. And those predominated in the classic categories that you might think, whether they be cardiovascular, chronic respiratory disease, or diabetes, and then hypertension. And then a good portion of these individuals were obese with the BMI greater than 30, about 50 to 52%. I'm going to spend just a minute orienting you to this slide because it's, you know, about a third of the bulk of our results. And so we collected tissues from, I think, 85 different anatomical sites. And those tissues were sort of grouped into different tissue categories that are listed along the left side of the slide there. So you can kind of see them: the respiratory, the cardiovascular, lymphoid, and gastrointestinal. And then within each of those categories, there's a specific tissues that we captured. And then along the top there, you'll just see a P whatever, and that's the patient number. So that's just each one of those columns will represent a patient. And then at the bottom of the graphic, you can see a time interval, or a DOI, which is the days of infection post-illness onset. So that's really the interval from illness onset to death. And you can see that we captured folks that died really quite early, within 14 days of illness. That's the early group. A middle, you know, kind of mid-tier, between 15 and 30 days after illness onset. And then another group that died, you know, 30, 31 or more days after illness onset. So we had a nice representation of the time frame. And I'm going to show you on this heat map is the RNA levels that we detected in the various tissues across these individuals, and I'm focusing on just the 11 individuals that we captured brain from. So we did 44 autopsies, but only 11 of those we captured brain. So for simplicity, I'm going to focus on the 11 that we captured brain. And the key for the heat map, shows that, a lot of our RNA, we used a droplet digital PCR assay that allowed us to normalize and compare RNA quantities within tissues, quantities within tissues, both across tissues within a single patient, but importantly, validly compare them across patients. So in this heat map, there's a lot of prep. A lot of virus is dark red, and not that much virus is light blue. Okay, so that's what the heat map is telling you. And so what you can see is that across all these 11 patients, that what we found of raw RNA across, more or less every single tissue that we sampled, and across all of these patients that are highlighted here. Importantly, the burden of virus was observed in the respiratory tract and arguably the cardiovascular system early post-symptom onset. So those that died early, but please note that obviously viral RNA wasn't limited to those folks. So if you look at P42 all the way to the right here, which I boxed, that individual died 230 days after illness onset. And importantly, this was an interesting case where this person had mild illness, had well-documented PCR confirmed illness during the early part of the pandemic, was fine, went home, recovered, and then came back months and months later due to an unrelated severe illness that ultimately resulted in their death. And then because we knew this individual had previously been diagnosed with COVID, we included them in the cohort. And indeed, we found viral RNA across all these different compartments. And these are the body compartments. But this next slide shows you same patients, but now focusing on 10 different regions within the brain. So again, these are those 11 patients, and the regions of the brain that we queried are listed along the y-axis there. And the main finding is that among 10 out of 11 of these individuals, we found evidence of raw RNA in their brain. And importantly, that included those all of the individuals in the late group. So including P42, who, as I mentioned previously, died 230 days after illness onset from an illness unrelated to their initial infection. So in order to kind of better, we had all this data, and so to better characterize the kinetics of and the burden of infection both within versus outside of the respiratory tract, what we did is we put all of our data points into a regression and worked with the statistician to create a regression line that told us burden of viral RNA, which is shown on the y-axis on a log scale, as a function of time post-illness, death post-illness onset, which is shown in a log scale on the x-axis. And on the x-axis, what I do is because again, it's the log scale, so you could lose track of this pretty easily. I just I denoted our early, mid, and late patient groups, so you can kind of track the time frame there. And what this graph shows, so respiratory tissues are in red, non-respiratory tissues are in blue, and the summary is that the burden of viral RNA is higher, about 100-fold higher in respiratory tissues compared with non-respiratory tissues, and that's shown by the intercept of those lines, obviously, and then what they do, but that the slope differs between the lines. And what the slope tells us is that it appears that degradation of viral RNA in the respiratory tract is actually more rapid than degradation in the non-respiratory tract. So you can see a negative slope for that red line is statistically significantly more negative than the blue line, which tells you a little bit about degradation of viral RNA in respiratory versus non-respiratory compartments. So Axel alluded to the fact that I have an affiliation with the Laboratory of Virology. I'm very fortunate, which is one of the NIAID laboratories out in Rocky Mountain Labs, and working with our collaborators there, For every sample that we found viral RNA in by our assay, we sent to them to independently, geographically different lab to use a separate PCR assay to one, validate our findings, but to two, query something else. And what that something else was the presence of subgenomic RNA. And for those that are kind of following this literature, subgenomic RNA is thought to be a molecular marker of viral replication. So you can do, a qRT-PCR targeting a subgenomic target that will tell you something about whether or not there's been recent viral replication. And that was an important question for us, and it's a high throughput assay. So we sent all these tissues to Emmy DeWitt and her group at RML, and they performed their subgenomic. And so what you're seeing here is our heat map. This is the same heat map I showed you before, but now overlaid on the heat map are these, and I can just kind of circle it here, but the subgenomic RNA and the key here are these sort of vertical lines, and it's a little hard to appreciate, but the boxes that have vertical lines cut through them are the samples that were positive by subgenomic RNA. And so the take-home point here is that on all of these early cases and one of our mid case out to day 18 post-illness onset, we found molecular evidence of viral replication in tissues, in respiratory and non-respiratory tissues across many different compartments of these individuals. But it wasn't just the early patients. It predominated in the early patients, but you can see here, patient P33, this is an individual that died 76 days after illness onset, and there was molecular evidence of viral replication in their tissues all the way out to 76 days. And so, again, two different labs, two different assays, and we did a Spearman correlation to say, well, how well, among those that were positive by both assays, how well did they correlate? And the answer is they correlated really well. They had a correlation coefficient of 0.91, and you can see the tight confidence interval there. And that's just to sort of further validate that what we were seeing was real. But as we all know, and we appreciate in medicine and science that PCR is one thing, but the gold standard is culture, is viral culture, and show me the money and prove to me that what you're seeing is real. And that's why we designed our study, so that we had an adjacent piece of tissue that was flash frozen, so we can go back and answer this question, because we knew, of course, it's going to come up appropriately so. And so viral culture, especially for CoV-2, is not a high throughput assay. It's a low throughput assay, and it's something that has to be done in the BSL-3 labs. And so we did, again, work with our colleagues at RML, and this was with Emmy DeWitt and Vincent Munster's group, and said, here are some select tissues. And we selected them based upon the amount of virus determined by droplet digital, but also our subgenomic assay, and said, go for it, see if we can, show evidence, the proof by culture. And so long story short, we sent them 55 different samples across different patients, different timeframes. And 25 of those 55 samples, we were able to grow virus in cell culture. And these are vero cells where the culture positive was determined both by cytopathic effect, but also a change in CT value by a PCR from what went in to what came out of the culture. So we validated this in a number of different ways. And, those 25 samples came from a total of 9 patients out of a total of 20 patients. We sent 55 tissues from 20 patients, and 25 of those tissues from 9 patients were positive. And the latest one that was positive was somebody that died 12 days after the onset of their illness. So we cultured virus in different tissues up to 12 days. And then we asked this question, is there a relationship between our cycle threshold value for our subgenomic RNA and our ability to culture virus from those tissues? And so we broke our samples into intervals of CQ or CT value. And so what I'm showing you here are the samples that had a really low CQ value, suggesting a lot of viral or subgenomic RNA. And in fact, those samples that had a CQ value between 15 and 20, every single one of those was positive by viral culture. And then as that CQ value went up by intervals of 5, then the percent positivity of culture appropriately diminished. And you can see that ratio here. And then the summary of this slide is that we grew live virus from not just many different respiratory tissues, which should be, somewhat expected, but also from multiple non-respiratory tissues. And those are summarized here. We grew them from the choroid and sclera of the eye, from the jejunum, from lymph node, from myocardium, from adrenal gland, and importantly, the heart, but not, the brain, but not yet. So when we did this on virocell culture, and we sent, Emmy and her group our high probability brain tissues. The first round we didn't get virus out of brain. So initially using standard virocell lines, this is a summary of what we found. But hold on, because we're going to come back to that. And then we asked a reasonable question. I mentioned that viral culture is a low throughput, high intensity assay that has to be done in specialized labs. And so we said, well, can we equate our ddPCR values, our copy number of CoV-2 RNA, can we equate a certain threshold to a positive viral culture? What's the relationship between the amount of RNA to a positive culture? And so we worked with our statistician to do a regression. And what you can see from this regression is that that the model using our data as inputs predicted that that a threshold of 758 copies, and we went after the two nucleocapsid targets. So copies of nucleocapsid gene per nanogram of RNA input into our assay predicts CPE, or a positive viral culture, with reasonable sensitivity, 76%, but really, pretty good specificity of 97%. And if you're familiar with these types of analyses, the receiver operating characteristic area under the curve just tells you how good your model is. So the closer that number to 1 is, means you got a really predictive model. So an ROC AUC of 0.887 is awfully close to 1, which means that we've got a pretty predictive model. And again, it's meant to just sort of say, if I just had a piece of tissue and I did my exact assay under our hands, the same primers and everything else, would that predict a positive culture to kind of help guide things moving forward? So it's one thing to find viral RNA, and to grow virus out of tissues, but that doesn't exactly tell you what cell types are affected. And so we worked with our colleagues in the NCI, and this is work by Stephen Hewitt, who's really an expert pathologist and quite facile in the different molecular approaches to characterizing presence of RNA and protein in tissues. And so this is an in situ hybridization assay that probes the spike gene in CoV-2 in various tissues. And this assay took quite a bit of optimization, because others have tried this, and there's some nuances to this. But what I'll tell you is that we conducted all the positive and negative controls associated with this assay, including applying this assay to pre-pandemic tissues across the spectrum of tissues that we, looked at to really kind of validate the specificity of our probe. And what we found with a specific probe is presence of viral RNA across many tissues and many cell types. And this is just a sampling of them. So here the ISH probe gives you a chromogenic readout with CoV-2 RNA in brown here, and you can see those brown cells that stain positive in thyroid, esophagus, spleen, appendix, adrenal, ovary, testes, and uterus. And in summary, we detected CoV-2 RNA in more than 30 cell types across 35 tissues. So we found it in many. We didn't find it in all cell types. So the dogma ahead of our study was that the virus does not get into lymphocytes, and we did not find any evidence of RNA in lymphocytes. There's a belief that and there are a couple of papers published on this, that the virus can get into monocytes and macrophages, perhaps via an FC-mediated mechanism. And in fact, we found viral RNA in monocytes, but really at a somewhat limited level, because the dogma there is also that while it gets into those cells, that it certainly doesn't replicate to a high level, and if it replicates, it's not in. I don't think there's any data to support true replication within monocytes or macrophages. Now, brain was another issue. So we applied our same ISH probe to brain tissues, and we found viral RNA probing the spike gene in multiple brain tissues across multiple individuals across the various timeframes that we studied, the early, mid, and late cases. But to say that there's virus, either viral RNA or viral protein in the brain is another leap, right? And so what we did is we took an orthogonal approach to validate our findings with additional assays. And so what I'm showing you here are a couple of additional assays applied to our brain tissues, which include immunofluorescence. You can see those images at the top, and those use an antibody that probes the nucleocapsid protein. And then at the bottom, what we have from these panels are two different ISH probes. So one, we probed the spike protein. Two, we probed the nucleocapsid, excuse me, we probed the spike gene. Two, we probed the nucleocapsid gene. And then D and J are IHC chromogenic, going after nucleocapsid protein. So really, depending upon how you count it, four assays using microscopy-based techniques to sort of orthogonally validate our findings in brain. So let me walk you through what we found. So panel A here is thalamus and viral protein nucleocapsid is pseudo-colored green. So and the Nguyen, which is a neuron marker, is pseudo-colored magenta, and then blue there is obviously DAPI to identify nuclei. So what you can see in some of these images, both with the white and the red arrow, is that you have viral protein in a perinuclear orientation. So this is within the cytoplasm, staining positive in neurons, so Nguyen positive cells within cerebellum. And then B, C, and D down here are the same patient, the same tissue, just using our orthogonal validation. So B and C, that's ISH for spike and nucleocapsid, and D is IHC for nucleocapsid, all confirming that in fact we were seeing what we believed we were seeing. And then E is a different patient and a different region of brain. This is now, actually it's spinal cord. So this is spinal cord from a patient. And again, you can see viral in a perinuclear pattern that is positive by immunofluorescence. F is just a higher magnification view of a neuron, and then G is a spinal ganglion from the same patient that is also staining positive for SARS-CoV-2 RNA. And then H, I, and J are also spinal cord from the same patient, but just with our other assays to sort of to support our findings. So that's sort of the microscopy-based stuff. I'm going to switch gears just a little bit, because one of the other questions that I mentioned that we had, I'm just checking time, I think I'm okay still. One of the other questions that I mentioned that we had was, is there any evidence that CoV-2 replicates and or evolves in different anatomic compartments? And so to try to address that question, we worked with the collaborator, Eli Boritz, who's in the Vaccine Research Center. He's a tenure track investigator. And what Eli is, one of his strengths is using a PacBio long read sequencing platform so that you can characterize major and minor variants of virus within a single sample. And so we had Eli apply his long read sequencing to tell us presence or absence of major and minor variants of CoV-2 in our different samples of interest. And so just to orient you to this a little bit, over here we have percent of single genomes, which gives you a sense of the percentage of the variants that we detected in our various tissues. So for simplicity's sake, we'll just start with this sort of middle one here. These two middle ones where you can see that 100% of the variants that we detected in these samples, and these were respiratory samples from a single patient. All these sequences are from a single patient. And these two middle ones are sequences from the left inferior lobe and the right superior lobe of this individual. And the sequence that we got out of those viruses were 100% the same in those two respiratory samples. Same patient, different anatomic compartments. We also sequenced samples from hypothalamus. You can see that at the top here, and then thalamus at the bottom. And for hypothalamus, you can see that we observed 3 different variants within that sample, and you can see the proportion of those variants over here to the right. And then, the colored graphics here are whether or not we detected a synonymous or non-synonymous mutation, and the magenta or the pink here represents the presence of a non-synonymous mutation that you can see at amino acid position 1219. This is a spike gene. This is just focused to the spike gene. So amino acid position 1219, you can see there's a G to a V substitution that we detected in 2 of the variants in hypothalamus, none of the variants in lung, and 1 of the variants, the predominant variant that was present in thalamus. And then importantly, you can see also here at this earlier position, there's a D to F change at the 80 position that was present in the lung samples, but not in the brain samples. And so what this is telling us is that we actually have distinct populations of virus in different anatomic compartments. What's driving those distinct populations? This is not designed to answer. It just tells us that what we found in lung, which we know, which we believe, which you understand, that is the site of entry of this virus, and that we know there's high level of replication, that what we find in lung is not necessarily what you find in other tissues. And that's what we observed here. And it suggests that there is, this was the hint that there was replication in these other tissues, that something was going on, and perhaps evolution of a virus in different anatomic compartments. And so when we had this data in hand, things had evolved a little bit as far as sensitivity of viral culture. And one of the things that became available was a modified viral cell line that expresses the human ACE2 and TMPRSS receptors, which it wasn't definitive, but there was a suggestion that this cell line might be more permissive for CoV-2 infection. And so we went back and tried again. And this is again with Emmy's group at RML, and we sent them thalamus. And this was, we had sent it previously. We selected it because it had a reasonably high level of RNA, both by our digital and droplet and subgenomic RNA assays. But when we tried it in traditional viral cell culture, it didn't work. Well, when we tried it in the modified cell line, in fact, it worked. We got CPE, and also we got a drop in our CQ value. But because we knew, again, burden of proof, you're growing virus in the brain, you got to prove it to me. So what we did was we already had the sequence of the variants that we had detected from the brain tissue itself using ELI's assay. And so what we did was we sequenced the virus that came out of our culture supernatant. And in fact, the sequence of the virus, which came out of our supernatant, which is shown here, this is the thalamus cell culture, matched the minor variant that was detected in the thalamus, and that's this sort of red arrow here, and it matched the major variant that we had detected in the hypothalamus, in fact, confirming that what went in is what ultimately came out of our viral culture, confirming truly presence of viable virus in this individual's brain. The next slides that I'm gonna show you are gonna make one point. So I'm just gonna tell you what that point is up front, and then walk you through the histopathology. But the main point of these other slides is that despite the fact that we found viral RNA in cells and tissues throughout the body, and persistence of RNA and really quite late for a number of months, and viable virus in many respiratory and non-respiratory organs, that despite that fact, that the histopathology and that the presence of inflammation, meaning infiltration of immune cells into tissue, was really isolated to the respiratory tract. So despite virus being in all these places outside of the respiratory tract, there was really largely absence of histopathology directly related to viral infections, so cytopathology, inclusion bodies, these types of things. And there was largely absence of infiltrating immune cells in all of these tissues that we found raw RNA. With that, then, I'm going to go ahead and give you a little bit more detail about the histopathology across different organs. And so this is just lung. And what we found in lung was basically what others who had come before us found in lung. Most of these patients were people who died from complications of COVID, 86%. There's 14% that died with, but not from COVID. These are folks that came into the hospital for some other reason that we incidentally, at that time, everybody was getting swabbed for COVID. We found presence of CoV-2, but then they died from something else. But most died with complications of COVID. Diffuse alveolar damage, which is the primary histopathologic feature in the lung, was detected in all but 18% of our cohort. And it was in different phases, which I'll go through. Many of these people had evidence of acute pneumonia. So this is like a neutrophilic infiltrate into tissues, which is often, a pathologic indicator of bacterial co-infection. So we actually did do a characterization of bacterial co-infection, which I won't talk about today. Many of these folks had hemorrhage or thrombosis in the lung. And this is just a graphic for, I'm a critical care ID doc. There's the critical care pulmonary docs that are quite interested in, the main feature of severe COVID, which is acute lung injury. And that has this correlate of diffuse alveolar damage, and diffuse alveolar damage moves from an exudative to a proliferative to a fibrosing stage. This is a long way of saying that that's exactly what happens. And as you get further out, the major pathology in people that were 3 to 6 months out was a fibrosing stage of diffuse alveolar damage. This is a summary of the findings in the heart. I kind of alluded to this, but 9% of 4 of our patients had some infiltration of immune cells into the heart. So but of that 9%, there was only one patient. Well, it's not there, but you can see it's listed diffuse lymphocytic myocarditis. There's only one patient that had a truly, diffuse infiltration of immune cells associated with myonecrosis that would then be characterized as myocarditis. There was only one patient, and that patient actually was an individual that had multisystem inflammatory syndrome in adults, or MIS-A. And we characterized that case, published it separately. Really fascinating. Side note, but we cloned the T cell. We cloned the T cell receptors in that patient's immune cell infiltrate in the heart, and there was a super hyper expanded clone that accounted for 50-some% of T cells. One single clone that accounted for 50-some% of T cells in multiple regions of this person's heart. So super interesting case, but an oddball, right? Like not the main finding. A number of these folks had acute microscopic ischemia. This is sort of like microthromboses that was present in the heart. This is lymphohistopathologic findings. There's this question in severe viral infections, and Ebola is probably a great example of it. When you have severe viral infections, oftentimes we see lymphopenia on a white blood cell count, and at least in Ebola, both in humans but also in tissues of animals, there's a correlate to that, which is that there's massive lymphodepletion within secondary lymphoid organs. And so we're curious. I mean, it is true that there is some degree of lymphopenia in severe COVID, but our question was, how well is that reflected in the organs? And so when you look at lymph nodes, lymphodepletion was absent in 78% of our lymph nodes. So lymph nodes, it's not that the architecture was normal, it's just that lymph nodes just hadn't, like disappeared. The spleen was sort of the opposite finding. Now, spleen is a little bit hard because it's one of those organs that post-mortem can become like undergo liquefaction and break down and that kind of stuff. And so the spleen findings are a little bit challenging, but most of the spleens, we found evidence of lymphodepletion. Again, the idea here is to kind of say, well, what's happening with an important component of the adaptive immune response within immune cell response within tissues? And we're working on that as sort of a separate effort. But this is a summary of our findings within the brain. The highlights here are that these were sick people. Many of them were in the hospital for a long period of time. And as a consequence of that, they had non-specific findings in their brain that, one might expect from critically ill people that had been in the hospital, the intensive care unit, had critical illness. And so many of them had evidence of hypoxic ischemic injury. Many had evidence of vascular congestion. And a couple had evidence of microscopic hemorrhage. But what is not listed here is evidence of direct viral cytopathic effect and or massive inflammatory cell infiltrate in the brain that one might, attribute to like a viral encephalitis or something like that. So the summary of our findings, CoV-2 burden is highest in respiratory tissues, but is widely disseminated in the body and brain, that the viral RNA can persist in multiple anatomic compartments up to, at least in our cohort, 7 months, that the virus can infect multiple cell types and that it can replicate in multiple tissues, including the brain, and that we observed a paucity of inflammation or direct viral cytopathology outside, an absence of that outside the respiratory tract. I just spent a little bit of time, just a second on this slide, a few couple minutes. I mentioned that this was a huge collaborative effort. We accounted for our collaborators under the moniker of the NIH COVID-19 Autopsy Consortium. I think most of the members of that consortium are listed here, just to name a few. So at the University of Maryland, I had a partner that really helped establish our network among the critical care providers in the region. That's Allison Grazioli, a very intelligent and directed and proactive critical care physician and nephrologist. Actually, she's dual trained in critical care nephrology. And then, members of my group in the critical care medicine department, this study was led. The first author is Sydney Stein. Sydney is a veterinarian who came to do a postdoc in my group and really took charge and did all of, her and Sabrina Ramelli, who's a postdoc, a PhD trained investigator. They did pretty much all of the droplet digital PCR across thousands of tissues, for this study. And then our NIAID collaborators, I mentioned Emmy and Vincent and Meet, who did the viral culture in Emmy's group. And then in NCI, they called him my work husband, Stephen Hewitt. So Stephen's a great guy. He's a pathologist in the National Cancer Institute, and David Kleiner is the other senior pathologist in that group. And then all of their trainees participated in this. They put the PAPRs on. We created this brain box to help reduce splash, and those guys just did an amazing job. And then a number of other institutes across NIH, and then obviously the clinical center admission staff. They would take my calls. They came to know me by name, every single one of them. I would see them the next morning, but I would talk to them at like 3 a.m. when we were doing this. So that's why we haven't done more. I could only do the 3 a.m. thing for about a year. But anyway, I think that's it. And again, I thank you guys for your attention, for the invitation, and I'm happy to take any questions.
I ask a question?
Please.
Yeah. So a few points. One is about the patient themselves. Thank you very much. This was a very nice presentation and a very unique group of patients. The fact that these are patients who died from COVID, as opposed to many patients who endure COVID and don't die. Do you think how applicable this is to other more wider population of COVID patients? And do you know if these patients were treated with antivirals or steroids or any other type of treatment that they receive these days, and how that could have affected the postmortem findings? Another question has to do with how many patients accepted, or how many families accepted for the post on these patients. Is this something that most patients would accept, or actually this is a very select type of populations that you eventually got a hold of? And
well, you better be careful, because I've got 3 of your questions in my head so far, but if you're testing me about how many numbers I can remember at once, I think it's about 3. No, no, I'm just kidding. You might need, if you have more, might need to remind me, but go ahead.
Well, probably the last one has to do more about the testing itself, and how sure you are that the RNA you're finding is in the tissue itself as opposed to blood vessels in that tissue.
Yeah, no, they're fantastic, all fantastic questions, every single one of them. So let me see if I can go through them one by one, and you may need to remind me. But the first is the generalizability of our findings to other populations that are, perhaps less old, less sick, so younger, healthier, perhaps vaccinated, so I think that's the first question. And the answer is we don't know yet, right? Because we focused on the patient population at a particular point in time before vaccines. It was really heavily weighted to older and sicker folks. But I will say that there is NIH funding under the RECOVER protocol that is, supporting, I think, up to $1 billion to understand long COVID or post-acute sequelae of COVID or past. And a portion of those dollars are to support autopsy studies that can answer the question you're asking about generalizability. And those projects are ongoing, both in the extramural community, and we are pursuing them to some degree within the intramural community as well. Your second question related to, well, what treatments did these folks get, and is there anything special that might have predisposed them to have the findings that we found? And the answer to that question is the individualized patient level data is all summarized in fairly good detail in a supplementary table within the paper. So if my answer is inadequate for you, you can reference to that. It's published online. It is open access through Nature on December 14th, so it is available. But these individuals got a hodgepodge. Some did get remdesivir, which is an antiviral at the time that was of interest in being studied. Many of them did get steroids as folks folks with critical illness due to CoV-2 now is the standard of care, as you likely know. And so, but also, and this gets to your third question, which is, was there something about patient selection that perhaps biased our sample? But what I'll tell you about that is that, which I think is reflected in the range of when patients fell post-illness onset. We had folks that came in and because of the diversity of hospitals, I mean, one of our hospitals was through Tidal Health. It's a health group that covers the eastern region of Maryland, which is a fairly rural part of the county, or which is a fairly rural county in Maryland. Is about 3 or 4 hours by ambulance ride. Those would be kind of like long rides through the night. But we had many number of patients that came there that were do not resuscitate, that really got little to no treatment, that died within a very short period of illness onset. And the findings that we observed, there was no distinct difference between those patients versus the patients that were hospitalized for a prolonged period of time. We had really a great cohort. We had a young person I mentioned was 6. years old that was really tragic case. This is a person that had a seizure disorder that they were born with, that had presented with status epilepticus and had a complication, was incidentally found to have CoV-2, had no respiratory symptoms on admission, had a clear chest x-ray. Matter of fact, had clear histopathology largely in the lungs, but we found virus disseminated throughout the body and brain in that individual as well. They died due to a complication during intubation to kind of move to propofol to control uncontrolled status epilepticus. So I don't know, but I think we had a representative sample. And then what was your last question? I told you I can only remember 3 numbers.
About tissue versus blood vessels.
Oh, thank you for your question. Yeah, so tissue versus blood. So we did address that question. So I didn't show the data here, but in every single one of these, well, not every one, I think there were about 3 that we did not have a perimortem plasma, or I should say perimortem blood sample, because for all but 3 individuals, we had a perimortem blood sample, which we then we spun down, we collected plasma, and then we also isolated PBMCs. And so we were able to query both the plasma and the PBMC fraction for presence or absence of viral RNA to begin to address the question that you're asking. The latest that we detected viral RNA in a plasma sample was, I believe, around day 14 post-illness onset. And remember, we had folks in our sample that in our late cohort that started at 31 days or more post-illness onset. So none of those people had virus RNA detected in blood. And as I alluded to with regard to the lymphocytes and macrophages, lymphocytes, there's no evidence that lymphocytes support infection of virus, or certainly, and there is evidence that monocytes support virus. So when we queried our PBMC fraction, we found really trace levels, trace to no levels of viral RNA in monocytes, and certainly not beyond the interval that I described. Suggesting that virus is not, trafficking in blood beyond really early post-symptom onset, and is not, meaningfully trafficking in any sort of cellular compartment. But the other part to address the question that you're asking is why we pursued microscopy-based approaches to confirm our PCR findings, to show using multiple microscopy-based assays that virus is not just in a blood compartment, but is in fact infecting epithelial cells and endothelial cells and fibroblasts and other parenchymal cells. So I think that's my response to your final question. Yes, please.
So although it's sort of assumed to be the case, in looking at the different cell types that were infected, did it correlate really well with these expressions?
Yeah, so the question, in case folks on Zoom couldn't hear, is how well the cell types that we observed that were infected, how well did that distribution correlate with the map of SARS-CoV-2 expression on cells throughout the body? And the answer is it correlated pretty well. But we actually also, and don't push me, somebody wants to be mean, they can push me, but we actually found viral RNA, by-ish in cell types that are not known to express the predominant receptors for CoV-2, the ACE2 and the TMPRSS2 receptor. But those are fairly ubiquitous receptors. They're on many cell types, and there have been efforts, the Broad Institute and otherwise, to sort of create these proteome maps to characterize that. We didn't put them side by side. Others have done that, to sort of say, that would have been an interesting graphic to say, this is our known distribution of these receptors. And this is where we found there was a paper before us that did that. So we chose not to replicate that. But most of the cells that were infected did express those receptors, but not all.
So,
go ahead, Peter. Yeah.
So there were 11 patients infected out of the 44 patients we had, because we had the brain on those 11 patients. So out of the 11 patients, how many were males?
How many were males? Oh man, I don't know. He's testing how well I know the data. I don't know. Not all of them. Some of them. The little girl was a female, but.
So the question is that you showed some data on testing.
Yeah.
So whenever we were looking at those slides going through, I saw that in the testes you showed some subgenomic of them, when you tested in their testes, from subgenomic RNA?
Yeah. Vivek's question had to do with the testes, and whether or not among all the testes samples that we evaluated, if we observed subgenomic RNA in testes in all of those samples, or perhaps what proportion of those samples did we observe viral RNA? And I think your question is getting at what we know about other RNA, enveloped RNA viruses, where both the testes and the brain can serve as an immune protected reservoir of sorts for the virus. To directly answer your question, we do have a table in the paper that summarizes both the average copy number of gene copies per tissue type, broken down by our groups, early, mid, late. So, which I don't have memorized off the top of my head for testes, but that is in there that says, hey, this is the amount that we found in testes among early, mid, and late, and it gives you the numerator, denominator for how many tests. And it also, that table also reflects not just the average amount, but the proportion of individuals where which is the exact question you're asking, the proportion of individuals where we had testes, how many of them were positive for subgenomic. I don't know the number off the top of my head, but it was not all of them. It was not all of them. Some we found just, RNA by ddPCR, and our subgenomic was negative. And your question gets at another thing, which, people are really driven to, and appropriately so, to say, well, what is the reservoir? And I don't have a good answer to that question, but what I can tell you is that different from Ebola, or at least, I don't think these studies haven't been done in Ebola, right? Like, this type of design hasn't been done. What we know about Ebola patients is if you go and test semen or you take a CSF sample in a subset of folks, you're going to find viral RNA. But what we found is, presence of viral RNA across, many cells and tissues far across time, months post-illness onset, both in known sites that are known to be immune protected and sites that are known to not be immune protected, which sort of raises a broader question, which is, what are the molecular mechanisms that allow viral RNA to not be sensed or responded by, intracellular pathogen recognition receptors? What are the mechanisms that somehow avoid an intrinsic interferon response or other cellular response that would result in clearance of viral RNA? What are the mechanisms that perhaps, contribute to failure of cells to process viral proteins and present those proteins on MHC class 1 and class 2 molecules such that the adaptive immune system can come in and clear out virus from those infected cells? Something's not working because viral RNA is allowed to persist. And I think, germane to your question is, what are the underlying mechanisms that allow for that? And it's important that and the answer is, I don't know, but it's an area for ongoing study. And the implications are that, man, we need to understand those mechanisms if we're going to hope to reverse them, right? Yeah, full stop.
Thanks. The other question is about, you mentioned about one risk patient device.
Mm-hmm.
Is it a child or is it a...
It was an adult. It was actually, so that case it's an adult. It's depending upon whose definition for MIS that you read, because this is a syndrome, and different bodies, the Centers for Disease Control, the World Health Organization, a number of different societies have come out with their own syndromic definitions for MIS. But this was a 20-some-year-old male, otherwise healthy male, who died during the early days of the pandemic. And he died of a clinical picture consistent with fulminant myocarditis, severe chest pain, had a cardiac catheterization, clean cardiac catheterization, serum troponin levels, a marker of myocardial injury through the roof, who ultimately arrested. They tried to transition him to ECMO to rescue him, and he died despite that. He tested negative for CoV-2 3 times pre-mortem when he was in the hospital. But because it was early in the epidemic, and you may remember this, there were a number of reports saying that in severe COVID, one of the manifestations is myocarditis, and there were some imaging studies and whatever. You know, we said, okay, well, we need to take a look. So we included that person into the cohort, and we found traces of viral RNA in the GI tract and in the lymph node, both by ddPCR and by ISH. But he had no lung pathology. We didn't have a timing, right? Like, we didn't know when he was infected because he didn't know he was infected. But by the amount of our RNA that we had, if we threw that amount up against the other data set that we had, the answer is his infection was a while ago, right? Like, that's what we kind of were able to glean from the amount of RNA in his tissues. And he's the individual that had truly, histopathologically confirmed myocarditis. And he's the individual that we sequenced the T cell receptor population in his heart, and he had a clonal expansion where a single clone accounted for over 50%. Now, again, I'm getting a little long-winded, but for that patient, because some of the questions that come up with that are, what are the mechanisms that are driving, like in this otherwise healthy young person, a fatal myocarditis, right? And perhaps, is there any relationship between what we saw in this case that met the syndromic definition for MIS-A, multi-system inflammatory syndrome, in adults, because he's 20-something? Is there any overlap between that and what we're seeing with these concerns about young healthy folks and that are rare, but with the vaccine complication? And so the answer is we don't know. But what we did do, and we published this, is we threw the sequence of his T cell receptor up against a database of T cell receptors that had been functionally tested against CoV-2 antigens to say, is there a hit? Does his match any T cell receptor that are known to have antigen specificity against CoV-2 antigens? And the answer is no. So, I mean, it's a small database, right? Like, there aren't like thousands and thousands of receptors in that database, but we didn't get a hit. But if you allow bioinformatically for a little bit of sequence variability, and by a little bit, I mean 10% sequence variability of the receptor that we cloned, or that we found, that we observed, and what was in this database, with 10% wiggle room, because T cell receptors do have, even though they're quite specific, you can have multiple receptors targeting the same antigen. That once we allowed that wiggle room, there were multiple CoV-2 antigens that our receptor was predicted to hit. Now, the reason that paper appropriately was published in, this is not to say anything derogatory about Frontiers in Immunology, but it was published in Frontiers in Immunology and not Cell, because we couldn't prove functionally that our T cell clone was truly CoV-2 specific, or perhaps had some cross-reactivity against a host, a human antigen in the heart. And so we're actually pursuing that as well. We're working with some of the folks at NCI that have expertise in cloning T cell receptors, and then we're working to clone this person's T cell receptor and then do some functional tests to assess antigen specificity, both against CoV-2 antigens, but also against host antigens. No, we had so we did do HLA typing on him, because that's required for us to do the functional assay that I'm describing. I don't have it off the top of my head, but he had a common HLA type, and so we have taken the different permutations of his HLA, MHC class 2 HLA alleles, and we're cloning those, or I should say transfecting those into a permissive fibroblast cell line, so that we can present diverse CoV-2 antigens on that cell line in the appropriate HLA presentation, to then test our clone TCRs to look and see if we can find functional evidence that in fact that these clones are targeting that have antigen specificity against CoV-2. Question.
Oh, excellent work. It set up a foundation for a lot of work.
Thank you so much.
So I'm interested on the brain. You say that you detected replicating viruses in the brain, but not virus isolation. And I wonder, because you were doing subgenome, yeah? So my thinking is, because it's missing virus isolated in the brain, subgenomic RNA can be using a genomic RNA that originally entered into the cells as a template.
Yes.
So did you try the negative sense is more direct evidence of replicating those tissue?
Yeah. So the question again for the folks on Zoom was about, really evidence, the burden of evidence that we have supporting live virus or replicating virus in the brain, and asking specifically about, subgenomic RNA, which we know is a replication intermediate. And, there are actually studies about subgenomic RNA, and there are multiple subgenomic RNAs across, different that translate different proteins of the CoV-2 virus. And we know that we believe there's, the current understanding is that those subgenomic RNAs are actually protected in this like double membrane vesicle, so that they might be able to persist in tissues for a little bit while longer, and that therefore, if you detect them, they may not actually represent, really recent viral replication. And so that is really not the gold standard for then saying you have viable virus. And maybe, because I know I said it initially, but I maybe didn't say it clear enough. When we first tested to try to grow virus in the brain, we failed. Like, in other words, in cell culture, we failed. We used a standard vero cell line. But when we found by our sequencing that there were viral variants in our brain samples that were not present in our lung samples, we said, oh, we got to look again. And so by that time, there had become a new reagent, and the new reagent were this modified vero cell line that express the human ACE2 and the TMPRSS2 receptor, that cell line, although I can't say definitively, but there's a suggestion that cell line is more permissive. So we tried again, and we in fact did grow virus out of the brain. We grew virus out of the brain, and we confirmed that the virus that we grew out of the brain was indeed the same sequence of the virus that we had detected from our RNA later preserved. So we have gold standard evidence that in fact that viable replication competent virus has the potential to be in the brain.
So have you looked at serology, kind of IgM or IgG anti-spike, and any correlations between viral replication or different areas, and then that humoral response?
Yes, so the question was, did we look at the humoral response, both IgM and IgG, across the individual, and was there any relationship between the humoral response and perhaps, and don't let me put words in your mouth, detection of virus in different compartments and or viral load across compartments? And the answer is, we did have plasma from most of these individuals. And on all of the individuals, we did look for IgG responses. In a subset of them, we looked for IgM responses, and those are all outlined in a supplementary graphic in the table. And the short answer is that, to be fair, there were nothing particularly surprising about those findings. So earlier on, there was like an expected kinetics of when IgM shows up, earlier relative to IgG, and that, in all of our folks that were 2 weeks or later into their illness, we absolutely detected IgG. Doing a direct correlation with, which I think is more, specific to your question, the titer of like IgG or IgM, and then relating that to the amount of virus. We haven't done that yet. I think we probably are powered to do that and to get a little bit of an understanding about what the relationship is between, the kinetics of the humoral response and viral load. But we haven't done that exact comparison yet, no.
Question is, were any of these guys that were really far out with 200 and something, the other was still PCR positive by standard PCR, because you did see in one of those guys siRNA?
Yeah. Yes. So the question was, did we isolate virus from testes? Sorry, the second part. Oh, right. Sorry. Thank you. Yeah. Yeah. Right. Right. So the second question was, in individuals that, we'll just say for simplicity's sake, and don't let me put words in your mouth, that were in the late category, did any of them have evidence of persistent viral shedding by standard PCR approaches, sampling the respiratory tract, whether it's a nasal swab or bronchoalveolar lavage, or that type of thing? My recollection is that we did not successfully isolate virus from testes, and that might just be a sampling issue. Part of the heat map that I showed you, there were a number of gray boxes, and those are boxes where the sample wasn't available. So, the way that the study evolved is that at the beginning of it, we started sampling, I don't know the exact number, but let's say smaller number, let's say 30 tissues. But as we started getting our PCR results back, we're like, whoa, wait a second. We increased our sampling. And so part of it may be a sampling issue as far as the virus culture. The second question related to, you know, and I'll change the question a little bit. But don't let me put words in your mouth. But you know, what is the relationship between detection of viral RNA by sampling readily available samples from the respiratory tract, whether it's a swab or a wash or otherwise? How well does that predict, you know, presence or absence of viral RNA in tissue? And the answer is that that many of these individuals were PCR negative by the time they came to us. Many, if you know, especially in the late group. And we've looked at this in a different population that's not part of this publication, where we know, and I'll just give you the example because this is in press as well. We had an interesting case. We work with some of the transplant surgeons from the University of Maryland, and a related question to your question is, when is it safe to take an organ from an individual who died? They had a COVID infection, right? Like at a certain interval before they died, they had a motorcycle accident or whatever, and now they're an organ donor. When is it safe to take that organ, a lung, and put it into somebody else? And so that's a clinical question that folks have struggled with, and that is still an ongoing question. And because we were working with the transplant surgeons from Maryland, we had a unique opportunity to at least address this in a few cases. And what we found is, and now the current recommendations for this are, it started with a swab negative here. That wasn't good enough. Because you could be negative here and be positive down here in the lower respiratory tract. So now the recommendations are, you got to be negative here, got to be negative here by bronchoalveolar lavage before that organ can be transplanted into somebody else. So we have a case that's being published, and we think it's pretty standard, is that negative, but then when we go into tissue and we query tissue with our ddPCR and we query tissue with our ISH, positive for viral RNA. So, the answer is that, you know, as you move further out in your infection, what you find from easily accessible samples is not really representative of what you're going to detect in tissues, which means that in order to understand this question of persistence, in order to understand the generalizability of our findings, both in people that died from COVID, but importantly, people that recovered from COVID, that have post-acute sequelae of COVID or long COVID, to try to understand what role does viral persistence play in this diverse manifestation, you know, clinical findings. We need to get tissue from these individuals. We need to get tissue. So that is something that we're pursuing. It'll be a small group, you know, for a natural history study where we're going to actually, you know, recruit folks to go and do biopsies, lymph node biopsies with our interventional radiology colleagues, gastroenterology biopsies that can be done relatively safely, to try to relate and understand, you know, in folks that have these constellation of symptoms that are very well characterized both clinically and radiographically, you know, is there a correlate as far as pathogen and host within tissue? So those are things that we, and I think others, are also pursuing.
You're now going to say that if you have COVID, you can't donate any organs. That's gonna be a real problem.
Yeah, and let me be clear. So the comment was if you have COVID, you can't donate an organ. I should have told you the rest of that story because the take home point of our paper was that even though we found viral RNA, that individual was followed for 6 months post-transplant on immunosuppressants, where they had serial sampling of their lower respiratory tract by bronchoalveolar lavage. And we did not find recrudescence of virus in their lavage, and that person did just fine. So I think, and again, it's too early to say, right? But these are the kind of data that we need to then make informed decisions. But, actually the story ends happier than what you described.
But you're finding COVID in all these organs, then turn around and say, cannot transplant.
Right, which would not be a very pragmatic approach, right?
All right. I think it is about the time. So thank you again for giving this great talk. And I just wanted to invite everyone. We will take the conversation over to the BSB where there will be some refreshments and we can continue our discussion. So.
Okay, thanks again.
So I'm just gonna delete your